Well, hello everyone. I hope everyone's happy the Euro Cup is over. Maybe some people are already celebrating or being sad. No, it's great to be here. Thanks for the invitation. Yeah, so I want to talk to you guys today about the insights is in the detail and sort of like the challenges that we face with biological data visualizations. And specifically, I want to focus on what these challenges mean for developing BioVis software tools, but also what kind of opportunities are out there to really create impactful tools. And yeah, the reason why sort of like that topic came to mind is because over the last 10 years, as I've worked on various different software tools for exploring biomedical data, I kept seeing sort of like reoccurring challenges, but also solutions that help us to get over those. Before I begin, I want to thank a couple people here that really helped me shape the talk. So first of all, thank you to Trevor Mance, who gave a lot of feedback and thoughts on the talk itself, who is also the author of AnyWidget, a tool that I'm going to show you later on, as well as some other tools that I'm going to mention. I want to thank Ashley Wilson, our head of immunology at OZ, who helped me understand immunology, as well as Arpan, who did a great job there too. And they also both provided valuable examples. And then I want to thank Nazar Abdenour also for providing feedback to the talk and creating a cool new widget called IPy Chat, which you'll also see later. So yeah, a little bit about myself. So I'm a computer scientist and a visualization researcher, mostly working in the space of like visual exploration of high dimensional and large scale biological data. And during my PhD, I've worked a lot with like genomics and epigenomics data. And now I'm currently at a biotech startup called OZ, where we're trying to digitize and unlock sort of like the human immune system by analyzing high dimensional single cell data. And so what we offer at OZ is kind of like a data-driven machine learning pipeline to discover high-resolution, but importantly also interpretable cell phenotypes from cytometry data. And so at a really, really high level, what that means is like the data that we work with is like cellular data of these single cells, and you have millions of those. And for each of these cells, you're going to measure, or we have measured sort of like surface protein expressions. And then the key question here is like, how can we cluster those cells into different phenotypes? And a typical analysis so far or before sort of like was able to uncover like broad cell types, like B cells, T cells, various flavors of those. But kind of what OZ brings to the table is like these high resolution clustering that you see here in the middle, where each of these colors represents a different phenotype. And not only that, we are also able to sort of like resolve these phenotypes and align them for comparison, which is what you see on the very right. However, without sort of like providing you with any methodological details here, and if we would just focus on these visualizations alone, you might come up with some questions like, how are we actually characterizing these cell types here? Like what markers defines a B cell, the T cell, and so on? We might also wonder, if I tell you like we have this high resolution clustering platform, like how do we actually get these clusters, right? Just it's nice that we can see them in a UMAP, but what do these clusters mean? And if we're able to resolve those, then how are we actually doing that? How do we get there? And so we actually really need more information. Just a single visualization alone isn't really telling the full story. And I want to show you another example, also from OZ. So here you see our dashboard. It looks maybe very familiar with people who have been here before from Devin's talk. So you see the abundance heat map on the left side. Each row here is a phenotype, and it's sort of like dot-like plot. If you don't know it, it's kind of a flavor of an upset plot. Each plus here means a marker is expressed. And then the heat map itself is showing you relative over or underabundance. And on the right side, you have a cell embedding plot of the same data. And now for this example, what we want to do is find A or multiple phenotypes that are differentially abundant for well or poor mobilizers. And all right, it's playing. So here we see you can interact with those. The first thing we want to do is just filter down the data so that we only look at excellent and poor mobilizers. So we're going to select those two categories from the list, and that will filter out a couple of columns. What we also want to do is to focus the analysis a bit more in the next step. Once the filtering is done, we're going to filter the phenotypes themselves down by only looking at CD33 negative and CD66B negative cells, so natural killer cells in this case. And once we've applied the filter, you'll see the heat map gets a lot smaller, so it's a little better to handle. There's still 600 phenotypes, so still quite a lot. But because we're applying a second filter to only look at the most varying ones, we really only have to deal with 30 at this point. Now, the next thing, in order to sort of perceive any pattern in this data, we want to kind of pull in the metadata. So which of these columns, in this case, correspond to an excellent or a poor mobilizer. So you see these two, the row on the top, which is like pink or blue. And now we're just going to cluster, or not cluster, but we're going to reorder the rows here by differential abundance. And that very quickly pops out a couple rows at the very top, where once I've moved the legend to the bottom, you'll see that there's sort of a cluster, or more red cluster in the top right corner, which is a phenotype that seems to be overabundant only in the cyan, the bluish columns, which are poor mobilizers. Now, why am I showing you all this demo? So even though we sort of like relatively quickly get to sort of an insight, and we can quickly find a differentially abundant phenotype, but that still requires us to A, know the data, we need to know the methods, we need to know what we're looking for, and we also can't really rely on a single visualization alone, right? So far, we've only looked at two, and the video keeps playing. We can also pull up the sample statistics. We can go back and actually look at the raw data to verify that the algorithm is really doing what it's doing. But so here you see we're pulling up the sample statistics. But eventually, to get to a really biological insight, you have to look at so many different interconnected plots. And this is not unique to OZ by any means. Over the course of sort of like my research career, where I've worked on different visual data exploration tools, all the way from like visually exploring data repositories over work on sort of like visualizing epigenomic data to the stuff that I've shown you now, so high dimensional exploration of single cell data. There's always this challenge of like insights are rarely in plain sight. You usually always have to dig into the details, and there's usually never a single plot that just says, oh, wow, okay, got it. And so for instance, when you explore data repositories, you're often looking for a single data set in a sea of lots of different data sets. If you want to understand genomics better, you're often looking, what we've just heard about in the previous talk, you're looking at motifs, right? But they're small compared to the entire genome. So you have these challenges of like, how do you perceive these patterns? And in the single cell case, we often want to identify rare phenotypes in a sea of lots of common phenotypes that do not change or might not be of interest, which we still need to see because we often don't have ground truth data. And so that kind of brings me to the thesis of my talk, which is that in BioVis, the insights are often in the details. And those details can either come in the form of analytical complexity. So you need to understand where your data is coming from, which is a function of whatever method you use to generate that data, and so on. Or it can come in the form of like small subsets of a large data set, like a small motif or motifs across like a whole genome. And so the analytical complexity here is that understanding the meaning of a visual pattern, what does it really mean when you see a cluster, for instance, in an embedding plot, or some kind of like other peak-like pattern in the genome? What does that really mean, right? And that is typically, obviously, it's a function of the data that you're plotting, but that data is a function of whatever method you ran, and that method's parameters are typically a function of the input data, and the input data is a function of the previous method, and so on and so forth. And so you have this whole chain that you ideally need to understand to get to an insight. And the other issue that I've already alluded to is like when your data set is very large, but the insights or the patterns that you're looking for are just representing a subset of that data, then it's very hard to visually perceive these patterns. Like here in that illustrated scatter plot, I put in like a couple very tiny yellow dots, but nobody will look at those at first sight because they're these big blobs that just catch our visual attention first. And so how do we resolve all that? And so what I want to go over in this talk is sort of like bring awareness to that general challenge in BioVis, which we've just done. And next I want to go a little bit sort of like deeper and talk a little bit about what is actually the role or purpose of visualization in biology. So what do we really want to achieve? And what do these two things mean for BioVis software tools? And then I want to close with like showing you some exciting tools that can sort of like make our lives a lot easier. And so generally speaking, what's the role of data visualization? You might have heard various flavors of sort of like a very similar definition. So like the goal of visualization in computing is to gain insight by using your visual machinery. And if you read through the other ones, you'll see the sort of like the commonality with all these quotes is like it's something related to insights, right? You want to gain or surface insights. But what actually is an insight? There are various definitions of those, like a deep understanding, or the ground truth, the underlying truth. The definition that I like to use is the bottom in yellow. It's not a definition, it's just a way of phrasing it. It's just unknown but useful information. And then in parentheses, if it's an actionable insight, then that information helps us to make a decision. Now, because it's really hard to actually quantify what information there is, especially in a complex situation like biomedical experiments, Min Chen actually proposed to not try and measure how well does your visualization tool really quantify or help users gain insight, but instead measure the time until they get information to sort of like complete a task. And so the key thing here is really like getting stuff done faster than sort of like what the time it takes you today. And so to summarize this, I would say the purpose of visualization is to help users surface and understand useful information fast, i.e. gaining insights. And so in the context of now biomedical research, what does that mean and how does visualization really fit in? So the classic case is we have some method which produces some data, we want to understand it, so we visualize it. Oftentimes though, it's not just, you're always contrasting data, right? Like you always have two conditions and you want to see, gain an insight by looking at the differences. So you're visualizing those. But then you, as I mentioned before, you have input data. Without understanding those, well, you can't really understand the output data. So you want to visualize that too. And I guess you know where this is going. You also want to visualize the method. So ultimately, you want to apply visualization frequently as much as possible throughout the process. And so in reality, that's obviously a chain, right? And you have like lots of different blocks that are connected, and you want to apply your tool not just in one location, but throughout that whole analysis. And so to summarize, the purpose of visualization for biology, again, is to help surface and understand unknown but useful information to ensure sort of like an efficient human-in-the-loop pipeline, analysis pipeline, and to have visualization be applied broadly. And that's sort of in contrast to the challenges that I mentioned earlier, where we have to deal with analytical complexity and large data. So why am I telling you all this? The whole point to talk about this is because it has an impact on what we need to do to really create useful and sort of like impactful BioVis software tools. And I believe there's sort of like 4 key principles that sort of like you have to work on or that you need to fulfill to really sort of overcome these challenges. And now I want to go through those 4 and give you examples along the way of tools that sort of like have accomplished this. And so the first is like the BioVis tool should be there where the computation is and where the data lives. And first of all, why do I think that is important? Well, remember from the definition, we said we want the purpose of visualization is to surface information fast. If we have a setup like this, which is often the case, you have a pipeline. I hope that's visible. There's a box around it. But your visualizations are external programs sitting outside of that pipeline. Then you're creating pain. Because if the goal is to make things faster, but you're telling people, hey, why don't you just exit out of your context and open up a new program and maybe reformat the data 20 times and then reformat it again to import it. Well, even if your visualization is then, I don't know, 10 times better than the previous one, it doesn't help because you spend 20 times more effort on just like using the tool. And well, there's this sort of like unspoken truth that bioinformatics is like, I don't know what the percentage is, but it's like primarily just reformatting data. And it's funny when you just look at, I search what's the most annoying thing in bioinformatics, you find this Reddit post and the most upvoted comment is like, yeah, you have to reformat data from one tool into another, into another, into another, and then you realize it doesn't work. Yeah, so if we do the same thing with BioVis, then we're defeating the purpose of building a new visualization tool in the first place. So we shouldn't do that. Instead, we want to apply it kind of like all directly to the machinery so that it's in the pipeline itself. And I think the best example here that like showcases the impact of this is the Upset app. And I don't know how many people know Upset? So it's like, all right, yeah. The original paper, I mean, was already great, right? It introduced a very efficient new way of visualizing set intersections, but it wasn't up until sort of like the R port was published that it really took off and had a massive impact on like the scientific community. And the whole reason why, in my opinion, is because you brought the tool where the computation actually happens, where the research happens, where the data is. Another example here is a tool we've worked on some years ago. It's called HighGlass. It was originally published sort of as like a web application for visualizing these large-scale genome interaction maps. And you can interactively compose everything, but it was bound to the web. It was like a web server. So if somebody else wanted to use it, they had to set up a web server and figure out all the stuff that you don't want to do. So later on, we turned that into like a Jupyter notebook widget such that you can just launch your HighGlass instance right where you're fiddling with your analytical code. And that made it a lot easier to just like providing better access to the actual visualization tool. The next piece I want to talk about is composability. And we've already heard Devin talk about that earlier with this tool Loon. But the idea is here, like, as I've mentioned earlier, why do we need this? Well, as I showed you in the OZ demo, and you all know from your own work experience, a single plot almost never works, right? We need multiple interlinked plots to explain the complexity of biology. And that means we need multiple tools. If we have a setup like this, where we have all the tools and maybe we can already apply them, but we still have to sort of like go back and forth, open one tool, go back to another one, do something else. They're disconnected, right? And so it's hard to correlate visual patterns between two different windows, for instance, or tools. And you need to go back and forth between the different, these blobs here. These droplets are supposed to indicate different tools. You have to go back and forth between those. So ideally, what you instead want to do is bring them all together, just compose them in one single interlinked view. That makes it a lot easier to just correlate patterns. And you also don't have to reinvent the wheel for every sort of like new system that you build, because you might be able to just use what somebody else has built before, if it is composable. And so, again, one really neat example here is Vitesc. So you can see it's a tool for visualizing spatial single cell data. And you can see there's not a single visualization. It's like a composite of various different plot types, and they can all be composed together. You can all like pull them up as you need them and work in the data with your data in sync. Again, on the right side, since HighGlass was ported to being a Jupyter widget, you could now also simply compose it with other Jupyter widgets. And one of those is Jupyter Scatter that I've built. And then Nazar, for instance, combined these two together. And now all of a sudden you can like explore the genome in a different manner. Yeah, the Vitesc is also a great example for being where the computation is, because you can also pull it up in like a notebook. And so here you see an example of like in sync exploration of the genome using an embedding view and the genome browser. And again, that's possible just because these two things can be composed together. And you don't have to sort of like re-implement the same idea over and over. The next thing, which is probably the most obvious aspect, is scalability. And here the reason why is, I guess, like obvious, like data is only ever going to grow, right? Like measurement techniques are just going to be better. We're going to accumulate more data over time. And what you want to avoid is having like a presumed context that is too small, because that prevents usage of your tool in the future. And here I want to give like a little anecdote for why you directly want to, if possible, go for like GPU rendering and tiled data. It's coming from OZ, so it's a fairly rendering-wise a very, very simple visualization. You just have dots. So we have a distribution here. It's a B-swarm plot of cell abundances. And the presumed use case initially was like, well, we're going to have hundreds of samples and we're going to have a couple of steps. All good. Up until all of a sudden you have 1,000 samples. And when you render this stuff with SVG, you're going to get stuck. Everything gets slow because your SVG DOM is just going to be too big. Like your whole app is going to slow down. So you can avoid this by just using GPU rendering, which is now easier than ever, thanks to cool new tools, which I'll show you in a minute. Again, two examples. Genome Spy is probably the smoothest WebGL renderer.
That's a good question. I didn't see a correlation there.
I think that was from the video. Sorry about that glitch. Yeah, so here are two examples that really handle scalability well. So Genome Spy probably has the smoothest, like, just WebGL renderer. It's not the best when it comes to data scalability. So there you might want to use HighGlass instead. But another really cool example here is Viv. So the image I'm zooming in here is 1.5 terabytes in size, and it doesn't use any server, just a web app. So if you can, you want to aim for that, because then sort of like your ceiling is much higher. You can now use Viv in lots of different applications. You might have small images, then it's fine, and you might never need like such fancy rendering techniques. But you can also get it to like terabyte size data, and it still works, which is great. And finally, maybe the not so obvious aspect, which I call like offering bidirectional interactions. And what do I mean by that? So typically when we build visualization tools, it's like, well, the visualization is for the human, right? So we want to interact with the visualization. We want to click somewhere, do stuff. And that's all fine. But as sort of like complexity builds up, as data gets bigger, it would be nice to have some guidance. And we can achieve guidance by like combining our tool, for instance, with some kind of like machine learning model that automatically identifies patterns. But then you want the model to also be able to operate your visualization. You want your model to also be able to select points, to zoom into a specific sub area of your plot, or do essentially whatever a human can do also. And so you want both sides to be able to talk to or use your visualization tool. And so why is that? Why do I think that's very important? Well, remember at the very beginning, I said one of the big challenges is that the insights is in the details. And so how do we surface these details? If the analyses are getting more and more complex, one way out, again, is to sort of like have machine learning based guidance in your tool. And if your tool allows bidirectional communication, you can actually accomplish that fairly simply. And so without that, it's always going to be up to the human to just like sort of manually interact with your visualization and surface all the insights. But if there are too many insights, or if the data is just getting too big, too complex, then you might miss stuff. And so one of the examples here is like a system we worked on some years ago. Again, this is from the HighGlass times, where we combined HighGlass with a machine learning model that you could actively train. And the idea there was like to build a peak finder in the genomic world interactively. And there you would go in, and the system would basically pull out views from HighGlass automatically, show them to you, you would label them, and then it would update sort of like the top track with like a heat map, so you could find similar regions in the genome. Another example that we've also heard earlier is like in the first keynote is Gosling. And with Gosling, you can basically configure everything, right? It has like a very comprehensive view specification that allows you to sort of programmatically get you to anything. You can even sort of like generate plot types. And you can use this for recommendations, but you could easily also build systems around this for data exploration, right? Where you say like, well, which region in the genome should I look at next? If the system can zoom in there, then it can pull out these regions. And to make this a little bit more interesting and show you something new also, I wanted to show you how you can put all of this together if you have composability. If your tool is there where your computation is and where your data lives, if it scales, then you can quickly put together intelligent systems. And so in this case, I was just like toying around with like a chat interface that helps me explore single cell data, but not just answer like sort of like in a natural language case, but is connected to my visualization tool and actually shows me and pulls up what I want to see. And just shout out to Nazar for building IPyLang chat here, because it's cool. I've used it in that demo. So here is the tool. It's just a prototype. You So it's not perfect. There are lots of glitches still, but it's fun. So you can go in, and those are two different systems. On the right side, you have a normal chat interface, which has nothing to do directly with the system on the left. On the left, you see actually a new visualization tool that's called comparative embedding visualizations, where you look at the two columns, two different samples, both are embedded with OZET's embedding technique. So you see they are aligned, these two plots. On the left side, you have a healthy tissue. On the right side, you have a cancer tissue. But you notice that the clusters are still sort of in the same location, which allows you to visually compare stuff. At the top, you have like a categorical visualization. So it's just showing you all the phenotypes that we found. And at the bottom, you see like a heat map that essentially shows you which of these phenotypes are relatively over or underabundant. And you can already do a lot with the system itself, but it still requires you to manually do all the work. But if we just link that with an LLM, we can ask questions about the data. But the cool thing is the system knows about the visualization, and because you can control it programmatically, if I, for instance, ask if there are B cells in the data, it's not just telling me, yeah, there are B cells, it's also selecting the B cells for me, and I see them. And that goes both ways. So I can also go in. Oh, did I click too fast? No. So I can still interact with the system. So I can still select any cluster or phenotype that I'm excited about, and go back to the LLM and just ask, like, what is the thing that I just like looked at? And beyond that specific implementation, the key thing here that makes this possible is just like the visualization tool offers bidirectional interactions. So the model can select phenotypes, the user can select phenotypes, and by being able to do that simple paradigm, you can like very quickly build like these intelligent sort of like interfaces. And it's kind of fun to do these kind of things because it's like so easy now. I mean, these days this takes like a day to implement with all the tooling that's already out there. And you can ask fun stuff because it's grounded in the actual data. You can ask like, well, what is a differentially abundant phenotype that they actually found in the paper? And so here, I think it's this one, the one on the right doesn't even exist on the left side. So by definition for these two samples, it's differentially abundant. But now I'm asking for like, is that playing? Oh no. Sorry about that, the video stopped working. Hope the next ones work. All right, that's too bad. But basically, it's pulling out the differentially abundant phenotype that was found, and it's pulling it out of the paper, and then basically just telling the tool, hey, zoom into these and show the user what those are. And that gets at this challenge of like, if there's so much complexity, we can use machine learning to sort of like help us surface the insights faster. But we can only do that if the tools that we have actually can talk to each other, right? So if the tool wasn't able to programmatically select any phenotypes, well, then I would still sort of waste time by saying, okay, so there is this phenotype, but I have to sort of find it myself in the visualization, which defeats the purpose of serving information fast. And yeah, you can also do other fun things like ask about, obviously, because it's an LLM, you can ask about biology itself, and you can also ask about the tool itself. So here I was asking what the color map actually means. If anybody's interested, I have a demo up. We can play around with this stuff after the talk. And yeah, so the key idea here is like, again, what you want is bidirectional interactions. So the user, and doesn't have to be an LLM, but you just want an API. But for this demo here, the key aspect is also it was grounded in the methodological background as well as it knew about the data. And then you want synchronized queries to the LLM by it also knowing what the view state is, and you want to get back natural and structured output to prevent a little bit like from hallucinations. But the key idea there is that you can only do stuff that's in a very constrained space allowed for your app. And so even if the natural language is like, it's not fully explaining you the situation correctly, it can only sort of like show you a view that's valid for your application, which is really powerful. All right, so how do we accomplish all this? Usually it sounds like it's like you need to do a ton of stuff to get to all these things, but it's actually a lot less complicated to achieve, like getting your tool into the environment where it needs to be, and having it composable, scalable. That's all like sort of like much easier to achieve these days. So how can we get there? For integration, the simplest solution is like try to get your system into Jupyter, because that's where most people just experiment. And make sure to use sort of like existing data standards, like support pandas data frame. Don't reinvent the wheel on like color maps or something. Just use matplotlib. And most importantly, use any widget, because previously it was a pain to get your visualization tool into that ecosystem, and now it's just like joy. And to bring that point across, I want to show you a very, very, very brief sort of like demo. Who here has heard of observable plot? All right, yeah. So it's a cool library by the inventor of D3. And the difference to D3 is primarily that it provides higher level charts. So you don't have to implement the low level like plotting stuff. It just gives you like, here's a scatter plot, and all you provide is like a data set, and you say, this is the X coordinate, this is the Y coordinate. The downside though is it's only living in the web on their platform, but that's not where my data is, right? My data is on my laptop, on my cluster, within my organization, but I have access to it in Jupyter. So how can I connect the plot library to my data? And this is all you need. So it's essentially all you need these days is like 23 lines of JavaScript code where there's not much happening. I'm just importing the library, and I have a plot function that just says whenever my model changes, just rerun the, here we're using a dot plot as an example, and we're saying like take the x-axis that is being provided to you from Python, take the y-axis, and provide a color encoding. On the Python side, it's even simpler. All we have to do is say, where is our JavaScript file? That's the first line there. And then the other 4 lines are just saying, well, I have 4 state properties. And that's all we need. And so when you execute this, here's a little demo. So I have, let's say, this is my assumed data. It's just random. I just instantiate the widget now. Just pointing out here, it's like these 4 state properties, creating the widget. There it is. And it immediately provides bidirectional interactions. So if I execute this one cell, it's changing the Y encoding. And just for fun, I created like a set of drop-down components here. And everything is interlinked and automatically works. And so you can, with that kind of setup, you can very quickly get your composable tools that work with your data. And so next, for composability, as I just mentioned, you can also like, again, any widget is like the way to go here, because it creates widgets and you can just piece widgets together very easily. But the key, sort of the underlying premise here, is like you need observable and declarative view state. And in the Python land, that's just tradelets. And in the JavaScript land, if you want to dive deeper, there's this proposal for signals, which makes it a lot easier to sort of like synchronize state. But again, the whole key idea here is you want state you can listen to, right? So if your component has some kind of setting and it changes, you want to be able to tell another component, okay, react to that. Like in the chatbot example, right? I put text into the chat box, and when that's entered, I want another component to say, okay, got the text, or change sort of like my encoding. Also, shout out to Nazar. He's going to give a talk on Tuesday about sort of like composable bioinformatics pipelines through open standards. So if that's something that interests you, like, please go check it out. On scalability, there are two aspects. There's the rendering scalability and the data loading one. For rendering, the obvious two technologies that you want to pay attention to are WebGL and the new cool thing that is WebGPU, which brings sort of like GPU-based compute to the browser. And I've mentioned earlier, there are libraries that make it a lot easier these days to just render stuff with on the GPU. And one of the libraries that I really like is called CandyGraph. It's like D3, but it's just automatically run this on the GPU. So if you use that, you never run into these issues where your DOM is just too big. For geospatial or just generally spatial data, DeckGL is really great. And if you're in the Python land, and you have like extreme size data, then Data Shader is a really nice option. Yeah, for data scalability, I think there are like two standards that are really emerging. Like one is Apache Arrow for tabular data that I feel like everyone gravitates to. And the other one is maybe ZAR for array-based data. And then I also wanted to mention DuckDB because DuckDB is like an in-memory analytics database. What do we need to, why should we care about this as visualization people? Well, it allows you to do sort of like aggregations on the fly really fast, which can be very useful, right? You have like a big map and you want to zoom in. You want to sort of like on a server side, quickly aggregate that data and send it back. And one framework here to highlight is Mosaic from Jeff Heer's lab, which is actually DuckDB backed, and it allows you to sort of like very quickly browse through terabyte size data by just saying, hey, database, give me just the data for my view that I need. And yeah, finally, sort of like on the bidirectional interaction side, there isn't really any technology or toolkit that you can use. It's just like a mindset change. So instead of like employing like an imperative rendering approach where you say you render, there's an interaction and you immediately re-render, you take this extra route by just saying, well, if there's an interaction, change a state property and then re-render. And the reason why you want to do that is because, well, then you can have an API that also changes the state. It's really a tiny, tiny difference. And so here, I thought it's like when you look at the top, that's like classic D3 code that you see somewhere. You do something, but the key thing is you have a mouse over and mouse out handler. And oftentimes you see like when you mouse over something, it immediately re-renders. It says like change the color of this line or of this dot or rectangle, whatever. But then the issue is how would somebody else, for instance, a machine learning model, do the same thing? Well, it can't unless it knows about your entire rendering process, which is like overkill. The simple solution is instead of directly re-rendering, you just say, well, set the hover state to this object. Then the renderer can figure out how to do that. But then also an external party can come in and say, like, change the hover state to that, or show me a tooltip, or I don't know, move a line from A to B. Do the same thing that the human can do. And that's it. Those are the 4 principles. I hope they were somewhat useful. Yeah, and now thank you for your attention, and I'm happy to take any questions.
Yeah, thank you very much for this amazing talk. There could be no questions from the online audience, but the audience is invited to come up and ask a question. Fritz, just walk towards, yeah.
Hi, thanks a lot. Very dumb question. Could you go back to the slide about integration tools? I missed that one.
Which one?
Integration. Yes, there we go. Okay, great. Thank you.
That was an easy one. I'll also happy to share the slides. I'll probably put them online somewhere later today.
Thank you. It was really inspiring. So I have a stupid question for you because maybe it's already inside the four principle. What about flexibility with respect to the input? So I wonder if this beautiful OZ that you show can, I mean, if it has any constraint to only show and visualize single cell data, or maybe the UMAP can come from whatever else.
Yeah, so the platform itself is bound to single cell data, but I mean, in an ideal world, what you really want to do is like have a completely composable system. I know it's not always feasible to get that, right? But that's sort of like what we should strive for. And yeah, due to like certain things not being open sourced, right? Like you cannot use other data at the moment. I know, but
That was the only bad part. Beautiful talk.
but in like from a technical perspective, you could, right? If you, yeah.
Thank you for very useful insights. I have two questions, probably dumb as well. So first one is because I'm an R user, so all of those tools or software you just said, is there any like kind of a counterpart like in R world?
Yeah, that is a very good question. So so you can have the same widgeting approach in the R world. And so for instance, I don't know if I'll find the slide, but Vitesc, for instance, was like a really nice example of like exposing also, where is it? Here it is. So Vitesc, I know, is also exposed as like an R widget. And R widgets are also just like they're just web-based components too. Jupyter is also just, everything runs on the web essentially. The only, I guess, slightly sad news is that any widget, I don't think directly works with R, which, exactly. So we should write a pull request or like a ticket for Trevor, and then it might be supported tomorrow. Yeah, I suspected that.
My second question is because we're also trying to build up in-house in company kind of a single cell atlas. So many of those data is it's not open source or public. So we're still waiting to, for example, to use those in-memory kind of storage or in-disk storage. So can you comment on so build those kind of a tool, the TileDB and also the DarkDB, which I mean, maybe just comment on their like advantages or maybe limitations.
Yeah, that's a little bit like sort of like outside my area of expertise. I know like you can there are subtle differences between TileDB and ZAR, which is on like read-write performance, but I think you can use either one of those. And I don't think the application differences are that big. You can also, there's a tool called Deep Scatter from a startup called Nomic, and they also have a tiling approach that uses Apache Arrow. And at the end of the day, I think you can sort of like get to the same product with either of these techniques. But the key thing is you need to have some sort of like data structure that either allows you to stream data in an aggregated fashion, or that offers tiling such that you sort of like are able to visualize all the data that you want, if it's like more than 20 million points or stuff.
All right, thank you so much.
Hi, very interesting. I guess this might be more of like a design question, but when you have something compositional, but also very interconnected, how do you make sure you separate everything, but also keeping it digestible and understandable? Would you have any tips on that point of view?
That's a good one. I think the key thing is that you organize your state dependencies in whatever wrapping application you have. It's sort of like a manageable fashion.
Mhm.
Another approach that I've taken before is sort of like do a hierarchical composition approach. So not trying to have like two tiny pieces. Like if you have 200 components, then maybe it like gets unwieldy. But so if you just like recompose and say like, okay, so here's one blob of like 10 components and another blob of 10 components. Within that, it's like sort of like manageable. The other thing I would say is like it then really depends on what type of observable state implementation you use. Like signals is really nice. It allows you to avoid, that's getting very technical, but you avoid a lot of like setting up callback handlers for things to change. And so if you embrace a more reactive programming approach in that scenario, that plus maybe like hierarchical recomposition, then I think it gets manageable. But that sort of would be my approach. I hope that helps.
I'll keep that in mind. Thank you very much.
Hi, thanks for the nice talk. I have a question regarding Jupyter, really. It's
Okay.
more of like an implementation question. So I like Jupyter a lot, but it often comes up that you get like very messy notebooks, right? So you run things out of order and all this stuff. And this is without the widgets now. I imagine that if I add in like 5 more widgets that are all interconnected, you could really get something very messy, right? And very error prone to the user, right? Is it a good idea to like use Jupyter in this way, or to like set Jupyter as the standard for all these widgets? Or maybe we need something more standalone, but then we have this other problem, right? Where
Yeah, I mean, if I had a little bit more time, I would sort of like, I think you would break this up further. So I do think if you want to get your tool, like a BioVis tool, into the hands of people actually using that, it's essential to be sort of like where the analysis actually happens. And that oftentimes the fiddling, which is also where you need data visualization the most, I would say, is in these environments.
yeah,
But the next level sort of is like, well, don't sort of like have your Vis tool just be a Jupyter widget. So this example with observable plot, for instance, is like, I think the ideal scenario, you have like a web component that's just a web component. You can pull it in any kind of web context. You can have an application. Then you can also pull it in, as I showed, into a widget. And then the widget is extremely lightweight. But then you can also sort of like build out a platform, for instance, and say like, in my platform, I'm showing the very same visualization. And the widget at the end sort of like just becomes a connector to the Jupyter world. Instead of, yeah, I would probably not recommend building your whole pipeline in Jupyter. The point was more like, if you really want sort of like hooks early on to use it in the exploratory phase, then I think it's a good idea to have it there. But I agree, it gets unwieldy. Yeah, there's always sort of the dosage matters, right? Like, I hope that helps.
yeah, no, thanks.
Actually, another kind of answer to that is that a lot of dashboarding frameworks are now accepting Jupyter widgets as components. But Fritz, great talk, and thank you for the generous shout outs. But really, my question for you is actually, given this, I like this paradigm of machine learning guided, discovery of insights. Do you see, this is kind of an open question, do you see ways in which the same kind of paradigm can also be used with the pitfall of exploratory data visualization, which is distinguishing in noisy patterns from true insights? Don't mean to put you on the spot.
Yeah, I think that is an interesting open question, and that there are a lot of angles to that. So one of the tools, next one. So Yeah, so one example, for instance, is for this, the left side, the high glass ML peaks kind of stuff. We used like a vision model to find peaks. And one nice side effect of that was the vision model was also averaging away sort of like certain types of pattern that a human wouldn't see. So in that case, it was nice because it wouldn't pick up patterns that you would otherwise not understand visually. At the same time, like I mean, it's a hard challenge to pick apart, like what is like a noisy pattern versus what is another one. There are lots of approaches, like you could do a broader sampling of whatever you found, trying to just see examples of whatever your hypothesis is true in other scenarios. I'm blanking on the name. There was a nice BioVis paper that just like, very simple technique, but you're basically just saying, well, let's take this example. You're pulling out AP. Don't just say, cool, I'm done. Let's maybe quickly have some sort of like walkthrough for the next 20 other patterns that are also being found to tell you whether that's true. Is that going in the...
Yeah, that's cool. Yeah, I'm almost thinking about like a hypothesis testing correction that becomes more and more conservative as you keep visualizing, sort of like protecting you against data sniffing. It's a very cool idea. Like
I know there's this eternal struggle between, at least I think us BioVis people and the statisticians who argue like every single time you're doing another test, you need to have hypothesis test correction, which quickly becomes infeasible if you look at too many things or you have too many dimensions.
Right.
I know it's a hard challenge. I don't know what the best solution is. What we always say to people is like, I mean, none of the stuff that you find in the visualization system, you should take for granted, right? If the tool tells you, hey, here's a differentially abundant phenotype, it's still up to you to actually run a test, whether that's true or not. You can't just say, great, got it. I got my target. Let's move on.
Yeah.
So I think that's a cultural important thing to realize, that just because a model tells you something also, it doesn't mean it's true.
But I think in your framework, you can build in sort of these models that help you, guide you away from making those mistakes, as well as finding insights. So I think it's